Right, welcome everybody to this event this afternoon. It's 3 o'clock. I just was checking my statistics beforehand, and I think something like 35 people have died from colorectal cancer already today, which is quite sobering. About 17,000 people a year die from colorectal cancer. So this is a very serious issue, and if we could diagnose it early, that number would be far, far reduced. So what this afternoon is all about is to demonstrate how in the UK we can bring together the private sector, the National Health Service, academia, the research institutes, and a whole range, particularly of diagnostic companies, including Origin, who have really put this afternoon together. And I should say that I'm on the advisory board, just to avoid any doubt. But if we can together solve colorectal cancer by early diagnosis, then we would have made a huge difference. And that is really what this afternoon is all about. Now, the order of procedure is that there'll be a number of presenters who will follow seamlessly on, one from the other. There'll be a break in the middle, and there'll be a networking time at the end. So there won't actually be time for questions to the presenters as they go through their presentations. But I hope you have a good afternoon and look forward to meeting more of you during the break. Hugo.
Thank you, David. We're just going to push across we've skipped a couple of slides. So I'm Hugo Lowry, I'm CEO of Origin Sciences. Welcome everybody, it's great to see so many people here today. So many of you, a lot of you in the room will know about Origin. We're all about a unique biospecimen being the rectal mucus. And the hypothesis started with the mucosal stream starts at your mouth, ends at your rectum, throughout the gastrointestinal tract. And you're constantly exfoliating material into that mucus. So we hypothesized if we could collect that mucus in the rectum efficiently, and then we could extract the DNA, that would be possibly be a very valuable and useful biospecimen. So as it says on the slide, the actual DNA we get out is 250 times more than you get from stool and about 750 times more than you get from blood. So I have got a device here, which I'll show you because it shows the simplicity. That's the OriCol device, and I hope everybody can see it. It's quite small from where you're sitting. It comes with a single-use syringe and a standard proctoscope. And so one of the beauties of the test is that you don't need any bowel prep. It can be performed by anybody who can perform proctoscopy, which is simply the art of inserting the proctoscope into the rectum. So patient can be done by a junior healthcare professional. So it can be done in primary, secondary, or intermediary care. And John Latham will be telling you more about the community diagnostic centres that we're now working with. So proctoscopes inserted into the rectum, introducers removed, device is loaded onto the syringe, like that, cap removed. And then we replace the introducer, and we simply inflate this membrane. It just touches the rectal wall, picks up the mucus on the outside, and then we withdraw it. So you can now see that the sample is safely contained back in the device. Everything's removed. The patient can now put their trousers back up. We add a preservative buffer to that, and then that becomes the shipping and storage vessel for that specimen. And one of the beauties is it's ambient, so we don't need any cold chain for that. Then that's currently shipped back to our laboratory in Cambridge, where Daniel will tell you more about the science. We've developed a pretty cool assay for extracting the DNA and refining it. So that's the device. Everything else will make a bit more sense when my colleagues present. Then a bit about Origin. So we protected our hardware assays and analytical systems. We're pretty good at clinical trials. We manage our own clinical trials in conjunction with the NHS. We've established supply chains. We've got other research going on with the NHS as well at the moment, which I'll tell you more about in a moment. And then we've also developed a strong market knowledge from around the world for deploying the test. So above and beyond the colon, we've got a study currently running look at pancreatic cancer and lung cancer. We've got some early indications that pancreatic might be something we can pick up through the rectal mucus, which would be very exciting. We've got, I've touched on clinical capabilities, and obviously collaboration is something we're really pushing forward for now. And then we've got some quite exciting publications coming up at the moment too. Then allele, so last part, is we've so encouraged and excited by what we've done with the rectal mucus. We're now looking at vaginal mucus too. So we finished our first study with allele, which was more about patient acceptance and clinical acceptance. And pleased to say that all 45 participants preferred our test to a speculum. So Megan is heading that up, but we won't be presenting any more on that today. So thank you to our collaborators. We've got 12 people here today who are going to be telling you about their involvement in our project, which is really exciting. Thank you to our investors. We have to pay for this somehow, so huge support, huge thanks to them. Our delegates, everybody here today. The Royal Institute, very fitting and brilliant environment for something like this, and how lovely to be in the centre of London. And then, and finally, we lost our much-loved colleague and friend only 10 days ago from Origin, Ling Lan Zhang. I know some of her family are here today. We lost her very unexpectedly, incredibly sad. And I'm sure Ling Lan will be looking down on us today, very proud of everything we've achieved. And I'm so sorry that she wasn't here to finish the journey with us. And I think that is over to Ian, who's our Chief Medical Officer. Thank you.
Thanks, Hugo. Good afternoon. I'm Ian Daniels. In my bigger hat, I'm an NHS colorectal surgeon. I've been doing colorectal surgery now for nearly 30 years. And in my other hat, I'm Origin's Chief Medical Officer. So I'm one of those unique people that nobody seems to want to shake their hand because they know where my hands go. And I can usually get somebody's trousers down in about 5 minutes. So, you know, I've been doing this long enough that at the end of the last century, we thought that by taking smears from the rectum and looking for cancer cells, we were cutting the edge of science. And yet here we are, this position now, where we're not only looking at the cells, we're looking at the fragments of the cells and all the rest of it. And medicine evolves. And as medicine evolves, of course, we have more and more complex tests. We have more and more diagnostic tests. I do an awful lot of colonoscopies. And I don't need to tell you the burden of a colonoscopy on the NHS, as you can see that on the news. Nor if you've ever had a colonoscopy, you tend to ask to come back next week for another. But of course, as we do all of these things, it's got to have clinical value and it's got to have clinical usefulness based towards the patient. It's about improving outcomes for the patients in terms of both survival or cure of their disease, but it's also got a health economic benefit as well. We're all aware of the pressures that the NHS is under. It's on the news almost every day. And hence, therefore, we have to have really big multi-centre, often multi-site, often multi-recruiting clinical trials. It's all about matching the clinical data to the science data and looking to see what is most useful for those patients and how we can adjust outcome and how we can change their care. Essentially, from my perspective, I'm really interested in research that is directed for and directed to patient benefit. There is nothing more saddening than explaining to somebody their colorectal cancer is at a stage which is beyond our conventional treatments. And yes, we can put them into clinical trials, but looking into the eyes of people with the disease and their families, etc., is extremely traumatic for those that deliver the care. So how do we transfer our ideas into clinical practice? Well, of course, at the end of the last century, I thought that the secret was the Vogelstein method, the adenoma-carcinoma sequence. All we had to do was stick lots of colonoscopes up to lots of people, look for the little cauliflowers, i.e. the polyps, remove them, and they wouldn't develop bowel cancer. Of course, it's not as simple as that, as we're now aware, and there are multiple pathways, etc. And not only that, we now begin to select out individual treatments. Some people still go to surgery, others are looking at chemotherapy, radiotherapy, immune modulating agents, all directed by radiology and care, and surgery itself has changed from making big holes in people's tummies to using robotics and very clever microsurgery. Of course, all of this time we discover new conditions and new diseases. We're looking to try and prevent people getting their bowel cancer, and those at higher risk, the Lynch syndromes, the polyposis syndromes, etc., we're looking to prevent ever getting to that stage of having their cancer. But as I said before, there are very few people that come back and ask for a colonoscopy next week. There's also, of course, the process that takes you through that. And by looking particularly in other conditions such as inflammatory bowel disease, where we're now beginning to identify individual pathways in individual patients as to which particular immune modulating agent to use, we really need a test that we can therefore direct their care and select and monitor their response to their treatment. And as well as that, of course, we have the mass of people who come with tummy problems. As Lord Prior said, you know, there are still the same number of bowel cancers being diagnosed per year. Survival is improving. But the number of people that actually are diagnosed with bowel cancer through the standard clinical pathway is about 3 in 100. 97 people have other problems. And the box that we used to put them all in that was called irritable bowel syndrome is now being divided up into multiple different elements. And again, by having some device that can look and see and a technology that can assess for what is it that's giving you your symptoms could have quite a revolutionary effect upon work and life, etc. So in doing so, we have to work with our science team. We have to work to improve outcomes by developing clinical trials, collecting the massive clinical data, the massive biologic data, following those patients up, biobanking that information, and then looking back at it in a few years' time. One of the huge advantages we have with our cancer cases that we've collected so far is, of course, we're following them up. People with a large cancer may not spread. People with a small cancer may spread. There's clearly a difference in those cancers. And I'm hoping that in a few years' time, Daniel's going to be able to tell me which ones I should be most worried about. And hence, I'm going to leave it to him to explain the very clever science behind this to a simple clinician that ideally wouldn't like to stick so many colonoscopes up people. Thank you.
Hi, so my name's Daniel Wise, and I'm the Chief Scientific Officer at Origin. And as Hugo and Ian have already introduced, we work on a novelly collected mucus sample. And what I'm going to try and do in this slot is, to some degree, demystify the scientific part that always seems to be, you know, we don't understand, and I don't think it's often very well explained, and I'm going to try and do a slightly better job of that today. And then also how we leverage this into a strategy where we can use this to deliver groundbreaking diagnostics and tests. So starting off with what our research development flow is. So it's trying to explain 3.5 years of work in 5 minutes is going to be difficult, but I can at least give you the flavour of what we're trying to do. So we have this very complicated mucosal sample. We have the advantage that we're extracting from the site of pathology, right? So we're taking a rectum sample, so we're looking at this as almost a localized sampling. It's also a very rich biospecimen. You might hear that referenced a few times today. And that's because it's very complicated. So you've got all different sources of potential biomarkers, from the type of cells to cell-free DNA to proteins, et cetera, et cetera. So you've got a whole bucket of things that you can work with for scientific analysis. So what we have to do is decide what's important for X application. For instance, colorectal cancer. What biomarkers do we want to look at? And the first thing is preserving them. Because if you don't preserve them, once they're collected at the clinic, by the time they get to the laboratory or your analytical assay, then you're in trouble. You might not have the information you need to try and design a test. From there, you have to remove it, and that's what these two are. You have to take it away from that complicated mix and isolate exactly what you need. And this is where we say separation and DNA extraction. And these are methods we've either built in-house or collaboration, or through collaborations, in fact. Jen will be talking about this later for the extraction, to then optimise the part that we want to look at, make sure it's pure and it's ready for what would be a downstream assay. Then in some cases we've got opportunity to increase the signal. So if the biomarker is not very well abundant and you don't know really if it's there or not, but it might be important for diagnosis, there's other methods we've integrated in the lab that can make this picture more clear. And then this goes into some kind of omics assay. So what we refer to this as is our diagnostic development platform, and it's a way in which we think we can efficiently develop tests that are integratable with each other and co-deployable. So the example that I'm going to give today is colorectal cancer. So we've followed this pathway, and we've decided what biomarkers we want to look at, how to efficiently extract them, and then we get to our omics. I'll cut to the chase on the omics. So the novel approach we've taken is not just looking at the human genetics and epigenetics, but also the microbiome. We have the advantage of our sample type. It's easy to look at both of these contingents. So in this triomic analysis, what we're looking at is canonical human genetics, so this is mutations. We're looking at epigenetic modifications, this is what's referred to as methylation, but we're also looking at the microbiome, which is, I always think of as an environmental sensor. So we combine this together to get a broader view on gut dysregulation, and then I'm just going to give a couple of teaser slides of data that we've generated over the last 2 to 3 months. So the first thing is taking each of your omics and then looking at them and saying, is there a clinical or diagnostic utility to each of the things we're looking at? Or what I sort of colloquially refer to as biomarker buckets, so whether that be mutation, methylation or biome. So the methylation alone is just one example we've got here, and what we see we get is that your controls, which is participants that haven't got disease, generally cluster to one group of this plot here, whereas the different types of position of colorectal cancer cluster to the other. And this was a staggering result for looking at a relatively small methylome panel, which is what we did relatively early on. And then we've also looked at biome and mutation individually. Haven't got enough time to run through them all, but then we started combining them, so putting mutation and methylation together, and then looked at another very important point, these little red guys here, which we're referring to as advanced adenomas, and this really comes into early detection, so precancerous lesions, something that might become cancer in the future, and the earlier you can detect it and remove it, the better outcomes that you can give. So as we saw that we were having lots of very interesting, useful data for a diagnostic purpose from each omic, the next thing was, what happens when you combine them together? How does this help? Is it the same? Is it additive? And as we started to put this together, I mean, as I've just tried to succinctly put here, it's more than the sum of its parts. So when you put them together, you're getting a different view of gut dysregulation, how the gut falls out of balance. And at this point, you start to get a better and more granular view on what's cancerous and what's not. And it's also helpful with doing some samples. It's hard to tell, but as you combine more different types of features that you look at, or hallmarks of disease, through different omic approaches, you then start to be able to more reliably put them in the appropriate category. So the results at the moment are very exciting. This is an example of one of the multi-omic approaches we've put together. And what we need to do now is move this work forward. So we've been targeting, which seemed incredibly ambitious at the beginning, a performance of 97% sensitivity and 95% specificity. And as a scientist, you're generally quite pessimistic because everything normally fails in the lab. And as we've worked on this and built more and more and more, we're incredibly confident that we're going to meet this target for colorectal cancer. And we're starting to think about, once we finish this off, what we can do next. But most importantly, we need to get this to the point of where it can be deployed and start making a difference to patients. So how do we do this? So we need to apply what we've already been built. We can expand on it, we can improve it, we need to apply it in larger numbers, and we need to apply it in the cohort which we look to develop, so we look to deploy it, so the appropriate clinical pathway. And what we really need in this case, we need the next big study, and that's what this collaboration was initially all built on, setting up this big study, and what expertise do we need to take it from a societal unmet need to solving a problem. And the first thing that we need scientifically is clinical expertise and support. And with that, I think that naturally leads over to Mr. John Lacey-Colson.
Hi. So I've got 15 minutes to talk to you about how we're going to journey towards a new gold standard in the colorectal cancer diagnostic pathway. So let's start. So the poet Wilfred Owen used the term obscene as cancer to try and get people to see the horrors that he'd seen in the World War I battlefields. And that resonates with the emotional feelings that the word cancer brings out in most humans. My area of interest is colorectal cancer. And every year, globally, we see over 2 million new cases of colorectal cancer, 42,000 in the UK and 350 in my hospital alone. Now, these are big numbers, but they're abstract. What isn't abstract is the number 1 in 17. And what that tells us is that one person in every row in this lecture theatre right now will suffer a colorectal cancer at some point in their lifetime. In fact, in all likelihood, somebody in this room has colorectal cancer right now, but they don't know it. The good news is that for most cases, colorectal cancer is treatable. And in common with lots of different cancers, if we catch it early enough and act quickly enough, we can even talk about a cure. This is the reasoning behind the National Faster Diagnosis Standard, and that challenges us to move towards a diagnosis or a discharge from the pathway of patients within 28 days of their referral. Now, up and down the country, trusts have created pathways to try and meet that challenge. So in my hospital, this is the colorectal cancer, suspected cancer pathway that we've been using for the last few years. On the left of the slide, you can see the patient journey. And unfortunately, what that involves is a lot of waiting and worrying. Worrying about symptoms, waiting to see your GP, worrying about your appointment with the specialist, waiting for your investigation, worrying while the result comes back. The GPs do an awful lot of work before they refer us the patients to make sure we're seeing the right patients. But in my hospital, we get between 500 and 600 referrals every single month with suspected colorectal cancer. And so every orange square that you can see here has to be created 500 or 600 times every month. It starts with a triage by a colorectal nurse specialist, and then some patients go straight for a test. Some patients have a telephone consultation with a specialist nurse or with a doctor. Some patients have a face-to-face appointment with a nurse or with a doctor, and then they're sent for an investigation. The investigation is then sent back to a named responsible consultant and waits until the consultant writes back to the patient and tells them what the next step is. Up and down the country, we're failing. We looked at our figures recently and found that we're meeting this standard in under 50% of cases, and that on average, patients are waiting 28 days before they actually get to their diagnostic test. So what is that test? Well, for most people, it's a colonoscopy. So about 3 years ago, I had emergency surgery for an appendicitis. Embarrassingly, it was diagnosed by my 12-year-old daughter at the breakfast table. The next morning, my colleague came up to me and explained that, yes, my appendix was a bit manky and they'd removed it. But actually, there was a bit of my bowel that didn't look quite right. Two of my colleagues looked at it carefully and they decided to give me the benefit of the doubt, so they didn't remove the bowel there and then, but in 6 weeks' time, I would need to have a colonoscopy. Now, that was a humbling experience. I've been referring patients for colonoscopy for over 25 years now. But what I realised is it's not about the mechanics of having the test. The patient experience starts the moment that someone tells you you need to have a colonoscopy, and it starts with that waiting and worrying. Not all-consuming worrying that's there in your mind all the time, but little thoughts that jump into your mind, little questions. Questions like, I wonder if there's anything wrong. Questions like, do I have cancer? Questions like, will I see my kids grow up? And then comes bowel prep. Now, we tell patients bowel prep is special medication to flush your bowels out so we can get a clear view, but it's a little bit more than that, really. If you Google the words Billy Connolly colonoscopy, one of the first things that comes up is a link, and he describes in great detail his experience of having bowel prep and then a colonoscopy. Now, I can't recreate his colourful description, obviously, but what I will say is that I had an evening where I spent very little time enjoying the comfort of a silent night mattress and quite a lot of time testing the flushing mechanism on the Armitage shanks. When morning comes, the effects are still evident, and you wait for some sort of continent window of opportunity so that you can be driven to the hospital, hoping not to leave some long-term evidence on the upholstery in the passenger seat. When you get to hospital, you're presented with dignity pants. These are a one-size that really fits nobody garment with a big hole at the back to accommodate the educated finger in your bum, which is then replaced by something which is about the same width, but 2 meters long. This thing then wriggles around inside you for the next half hour or so, while the endoscopist negotiates the twists and turns of your intestines. Now, the outcome of this can be categorised according to a traffic light system. I was fortunate in that I was green, which is either normal or a very minor abnormality which requires no further follow-up. Amber is a benign pathology which can be managed according to national guidance, and then red is obviously a cancer. So what do those numbers look like? Well, we need to do 25 colonoscopies to find one cancer. For every 100, we find about 4 cancers in our hospital, and about 12 people have a significant benign pathology, but 80 to 85% of patients have essentially nothing wrong with their bowel. We see 100 patients every 6 days. That's what 500 a month looks like. And every year, over 5,000 patients are discharged from our colorectal pathway in our hospital. That's 5,000 essentially normal colonoscopies, some of which will have complications. Our local health economy spends between 3 and 5 million getting to this point on the colorectal pathway. And nationally, we spend about £1 billion up to this point on the colorectal pathway alone. We spend more generating normal clinical results than we spend on the entire treatment costs of all the patients diagnosed with colorectal cancer on that same pathway. And that alone is enough to make us pause for thought. So what's wrong with this experience? Well, it's awful. We're doing different things for different patients who are waiting and worrying. They're competing in acute hospital with patients undergoing emergency care. They sometimes get sent for the wrong test. It costs a lot of taxpayer money. For the staff, it's demoralising. We're not able to give the service that we want to be able to give. And it does our trust, reputation and financial damage when we miss our targets. So FIT has been introduced to try and reduce this problem. But with the mitigations for the sensitivity and specificity of FIT, we haven't seen a big drop-off in the number of referrals. So most patients still need to have an appointment with a senior clinician. Most patients still need a colonoscopy, and it still costs vast amounts of money. The system struggles to cope. And patients who are not on this pathway get pushed aside and can wait over a year to be seen. So with the TRIOMIX study, we have the opportunity to improve both the pathway and to think about a new test. So with the pathway, the first thing that we're going to do is move the assessments, the outpatient assessments, out of the acute hospitals so that patients are not competing with the emergency patients, with the ambulances queuing outside the hospital. We've worked with our local care board, and we've got clinical space in a community diagnostic centre, which is an entirely elective hub. We've already started running clinics where patients are seen by senior nurse specialists, and we've made 5 appointments to these clinical nurse specialist roles so that over the coming months we will be able to offer up to 600 new patient appointments every month on this pathway. The patients will have a detailed assessment and get sent for the right test. The test will then go directly back to the clinical diagnostic centre, and our ambition is to turn round and achieve the faster diagnosis standard by day 13 in 90% of our patients. That is a vast improvement on what we're doing now. It's better for our patients, it's better for our staff, it's better for our department. But we are still in the situation where every month we have over 500 patients and we don't know who has a condition which needs treating. So these patients still have to get sent for their colonoscopy so we can identify the pathology and give the right treatment to the right patient. And this is what we're doing to overcome that. So every patient who attends the clinical diagnostic centre will be invited to give an OriCol sample. Now the science. So we know when we look down a colonoscope that cancer looks different. We know that you don't just suddenly get a cancer, but there are pre-malignant areas which also look different, including polyps. We've known for decades that those macroscopic differences are mirrored with subcellular abnormalities. Abnormalities in terms of genetic mutations, abnormalities in terms of epigenetic phenomenon such as methylation, and even changes in the microbiome, the type of bacteria which are found in the bowel in and around cancers and precancerous polyps. We know that the colon is coated with mucus and that in normal health, as the bowel lining is regenerated, the normal cells pass through the mucus into the rectum and are flushed down the toilet in the normal manner. We know that if you've got abnormal cells from cancers and worrying polyps, they travel in the mucosa and they are lost in the same way. And as Hugo has demonstrated, we have a simple, safe and quick test which can be done on any patient in any outpatient clinic, and we can collect a sample of that mucus. That sample can then be sent off, and there is now the technology available so that we can measure the genetic abnormalities, we can measure the epigenetic methylation, and we can look at the microbiome changes. If in doing that we can find a fingerprint which identifies those cancer cells, which identifies those pre-malignant polyps, then we would truly move to a new gold standard. We would be able to change colonoscopy from being a maximally invasive investigation, which nearly everybody has to have, to being a comparatively minimally invasive part of a treatment pathway, which those at greatest need will have easier and quicker access to. That would be a new gold standard that the entire world would wish to adopt. The difficulty is the NHS can't do it on our own. We need access to people who have logistical measures in place so that they can ship the samples to laboratories that have access, easy access to high volumes of cutting-edge technology so that they can measure the genetic mutations, the epigenetic methylation, and the microbiomal changes. And that's why we need to collaborate with industry. And that is, of course, what today is about. And that's what the rest of this afternoon's talks will be on.
So I want to make just two thank yous, really. Firstly, to Hugo and his team, Ian, Daniel, Archie's over there, who I think have put on today, a really remarkable event today. I think for a small company to get this kind of collaboration from so many companies, whether it's as big a company as IBM or some of the smaller companies, is truly remarkable. And if we can deliver a colorectal cancer diagnostic, and maybe in time a screening test as well, which is easily accessible and which has the kind of specificity and sensitivity that Daniel mentioned in his talk, 97% sensitivity, I think, and 95% specificity, because we've got to have a small number of false positives. If you've got a lot of false positives, it kills it from a health system's point of view. And if the sensitivity is not there, it sort of kills it from the patient's point of view. So you've got to have both. It's invidious, I think, to pick out any one other person for thanks, but I am going to pick out one other person, who's John Lacey Colson, sitting here on my right, from Shrewsbury and Telford. And I want to pick him out because I know how hard it is to drive innovation and a change in the patient pathway in particular within the NHS. It is incredibly difficult to take on all those vast battalions who have an incentive not to change. So I think to have got this through Shrewsbury and Telford, through the CDC, the Diagnostic and Treatment Centre, there is a phenomenal achievement. If we can now then push that out through Manchester and other parts of the NHS as well, it will be a fantastic, fantastic progress. So on behalf of everybody here, I think, thank you, Hugo, for putting this on, and thanks to Origin for bringing everyone together. Thank you very much.